chromatin architecture. And I'm going to start by doing something a little bit different than we normally do. I want to start with my acknowledgements first. So, because this project absolutely would not have been possible without the people on this slide. And first is, of course, my funding agencies. The John Fetting Fund, I want to thank John Fetting and Vered Stearns for believing in me in this project and really giving me the money to start this work. Because I had zero data and it was truly just generation of preliminary data. And of course, the Cigarette Restitution Fund at Hopkins with Norma and John Grootman also for believing in my project. And the data that we have to share today is from that funding. My two technicians in the lab, Stephen Brown and Kelsey Ritter-Gordy, who have been relentless and very consistent in growing the cells that have been treated with alcohol for months and keeping them, happy and uncontaminated and growing well for months is not an easy task. And then last but not least, of course, are the bioinformatics team that I have worked with for the last several years has been a true collaborative effort. And it's just been wonderful to work with this group. They're members of the Experimental and Computation Genomics Core, and Leslie Cope is the bioinformatics director. And Leslie and I have worked together for many years, and he's on our call today, and I appreciate that. And Leslie works with Mike Considine, who's a senior biostatistician, and they've done so much of the analysis with the methylation and the bioinformatics data that I'm going to talk with you about today. And of course, Cornell Schubel, who's a laboratory director. Cornell was actually a postdoc in our lab group many years ago, and went on to do some work at the NIH, and is now back here at Hopkins, and we're glad to have him as a director of this ECGC. And Anuj Gupta, who's a bioinformatics analysis, who has also worked with me and helped me with the alignments and the RNA-seq data and so much of the data I'm going to show to you today. So I just wanted to point this out in the beginning because these people have been amazing in helping move this project forward. So, being that this is World Cancer Day, as my husband Kellogg Schwab reminded me, globally, there are more than 2 million new cases of breast cancer reported annually. The United States alone has more than 500,000 new cases every year. The worldwide prevalence is approximately 6.8 million cases. Although many risk factors for breast cancer are not modifiable, understanding the role of the factors that can be modified and altered is really critical. So alcohol consumption is a modifiable voluntary behavior that consistently predicts for a higher risk of developing breast cancer. And I wanted to show this photograph by Matushka, who's a really very excellent photographer in New York City and an artist. Back in 1993, she developed breast cancer. She was 37 at the time, and her mother developed it when she was 41. Matushka took a picture of herself and her mastectomy, and it was on the cover of the New York Times in 1993 when I was doing my postdoc work. And I have to say, this was the first picture I'd ever seen of a mastectomy. And she's done it so incredibly well. Well, that photo made the Life 100 photographs that changed the world. And I like to point that out. Matushka really just moved forward the whole acceptance of what women have to go through when they have breast cancer. And I liked her. She's alive and well today. So I wanted to feature her. Now, a 2018 study found that among avoidable risks, alcohol consumption was the third highest cause of cancer after cigarette smoking and excessive body weight. The study found that among adults 30 years old and older, alcohol caused about 87,000 cancers in 2014, or about 5.6% of all cancers, which is, pretty shocking. And according to CDC, alcohol consumption raises the risk of 6 different kinds of cancer. And that even light consumption, less than 1 glass a day of alcohol, can heighten the risk of contracting esophageal and breast cancer. Now, esophageal you can understand, and the esophagus is coated with alcohol. Well, why breast cancer? And actually in 1987, so a long time ago, before some of you were even born, the New England Journal of Medicine published 2 reports about alcohol consumption and breast cancer risk. In the two reports, both prospective cohorts, alcohol consumption, even at modest levels of intake, was associated with the risk of breast cancer. Now, I didn't know this, and most people are unaware of alcohol's carcinogenic risk. And I only became aware of some of these studies because, believe it or not, I was asked. I was interviewed by a magazine for some questions they had about alcohol and breast cancer risk. And I started looking in the epidemiological literature, and I started coming across all this evidence, which was shocking to me. What's also kind of scary is that evidence also shows that possible early indicators of breast cancer risk, such as benign breast disease and increased breast density, are associated with alcohol consumption. So here you can see an excised fibroadenoma, which is mostly fibrocystic tissue. It's a benign tumor. They can get quite large. Fibroadenomas and fibrocystic disease is common in a lot of women, can be a precursor for developing breast cancer. And here's a fatty breast that is actually very easy to image. You can see a very small cancer. But if you have fibrocystic disease or dense breasts, it can be very difficult to even see large cancers. So I found it really interesting that alcohol might contribute to this fibroblast behavior. Now, the World Cancer Research Fund and the American Institute for Cancer Research has showed in a 2018 update that based on 16 prospective studies of premenopausal women and 34 with postmenopausal disease, alcohol consumption was the probable cause and a convincing cause for premenopausal and postmenopausal breast cancer. So it affects all women, all ages, premenopausal, postmenopausal, all types of cancer. The meta-analysis showed that a 10-gram increase, and 10 grams is less than one drink, which contains 14 grams. A 10-gram increase in alcohol consumed per day on average, the risk increased 5% among premenopausal women and 9% among postmenopausal women. So in this graph, you can see that as you get older and you drink more, the more you drink, the more your risk goes up, especially with age. What's a standard drink? Well, I think most of us will be surprised to learn that it's only 1 bottle of beer, or 1 5-ounce glass of wine, 3 ounces of sherry, maybe a shot of liquor, and that the drinks in the cups that we'll probably be having during the Super Bowl or other parties are more than a standard drink. So keep that in mind. And although light alcohol consumption, less than a drink a day, does not significantly increase other types of cancer in women, breast cancer risk is increased by 4 to 15%. And this association is highest with estrogen receptor positive breast cancer, but other breast cancers, other types of breast cancer are also involved in this risk. What do 5 ounces of wine really look like? Well, it can be very deceptive according to the type of glass that's you're served. And you may be in a fancy restaurant, you get this really nice red wine glass, and there's a little bit in it, but it actually can be quite more than you think. So just be aware of the type of glass and how much alcohol you might be really drinking. So in the US, there's approximately 4 to 10% of new breast cancers that are attributable to alcohol. That's a lot, more than I realized. In Australia, regular consumption of alcohol accounts for 12.6 and 6.6% of premenopausal, postmenopausal breast cancer. And in the United Kingdom or England, it accounts for 12%. So several years ago, there was an effort by a researcher at Hopkins, as well as others, to put warning labels on bottles and cans of alcohol. And in this article by Hillel Aaron, he states that there's a broad scientific consensus that alcohol is a carcinogen, and that even moderate drinking increases one's chances of getting cancer. And he's quoted as saying, there's a lot of things that give you cancer out there, and alcohol is right at the top of the list. He says, I'm not advocating for alcohol to become illegal or even to be treated like cigarettes, but people have the right to know accurate information about how alcohol affects their health. So a letter was sent to Steve Mnuchin in the Trump administration, warning that saying that they wanted to post a label saying, according to the Surgeon General, consumption of alcoholic beverages can cause cancer, including breast and colon cancers. And of course, this went nowhere and nothing was done. So I won't talk anymore about the Trump administration. But you know, the alcohol lobbying industry is probably even more difficult than the tobacco industry as far as trying to express the dangers and the risks of alcohol. So despite a consistent association between alcohol consumption and breast cancer risk, the underlying genetic and epigenetic mechanisms remain unclear. Now we know that environmental factors and host factors and diet, lifestyle, alcohol can influence gene regulation and epigenetics and chromatin architecture. So epigenetic regulation of gene expression can act during differentiation of somatic cells in response to these environmental cues and stressors I just mentioned, passing on these modulations to the offspring, to, you know, us, to children, and they call it epigenetic inheritance. So what your grandparents ate and drank may have generated epigenetic marks that you have inherited. And if you want to know more about that, all you have to do is read the alcoholics type of and alcohol syndromes literature. And you'll find that alcoholics have inherited gene expression signatures that run in their family. So what is epigenetics for those of you who might need a little refresher? It's a stable heritable phenotype resulting from changes in a chromosome without alterations in the DNA sequence. There's three main types of epigenetic alterations: DNA methylation, which will be the main focus of my talk today, histone modifications, and also transcriptional regulation, such as with microRNAs. Now, silenced genes or condensed chromatin are associated with methylated DNA, methylated cytosines, and deacetylated histones. During when this chromatin opens and becomes more active, it's associated with unmethylated cytosines and acetylated histones. So DNA methylation can contribute to the stability of gene expression and can establish a silent chromatin state. Cytosines that precede a guanosine are often often methylated at this 5-carbon position. So the DNA methyltransferase family of enzymes take the precursor SAM and use that as a methyl donor to put a methyl group on the 5 position. Now, if you have these CPG islands, these lollipops represent cytosines, and an island would be a cluster of maybe 200 or so. If they're in the promoter region and they're unmethylated, the gene is usually active. So unmethylated promoters are associated with active gene transcription. But in cancer and other diseases, when methylation may be aberrant, the methylation of these CPG islands in the promoter can repress gene expression. So that's why it's important to understand methylation and how it can affect gene expression. And my student gave me this cute little comic, which will help you remember what methylation does. Oh, hey, how have you been, methyl? I have this great idea for a protein I want to make. It's going to be so cool. Just you wait and see. I'll have all these amino acids in a highly complex structure that'll make it. And then the methyl group's telling it, shh, silence. Now, I'm trying to see the top of my slide here. What other factors can regulate DNA methylation that we know about already? How can alcohol regulate DNA methylation? So we know that S-adenosylmethionine or SAM levels are reduced as a result of alcohol-induced reduction in folate and the inhibition of methionine synthase. At the same time, SA here inhibits DNA methyltransferases and leads to global hypomethylation. So we had some evidence from the literature that the metabolism, the effects of alcohol metabolism could affect DNA and as well as hypomethylation of histones and other proteins as well. So alcohol metabolism can exert epigenetic effects through several mechanisms, including increases in the NADH to NAD ratio and the formation of ROS or reactive oxygen species and acetate. And you can see here that ethanol can be broken down to acetaldehyde and then to acetate, which can go into the circulation And your results can be acetaldehyde addict formation, which can include DNA damage, increased reactive oxygen species, which can cause a lot of oxidative damage, and increases in the NADH-NAD ratio, which can definitely change energy metabolism. Now, what I was concerned about was this, of course, occurs in the liver, but how is it going to occur in cells in a culture dish? Well, there is evidence from studies that acetaldehyde, a carcinogen, can concentrate in mammary cells following a single exposure. And in an animal model, acetaldehyde accumulated and persisted in higher concentrations in breast tissue than in blood. So that made us realize that some of these metabolic effects could also occur in cell culture. So we decided to, as one of the experiments, to try to address how does alcohol affect the epigenome in breast cancer or in breast cells. We started to culture a normal human mammary epithelial cell line. It does not form cancer, but it's been immortalized with H-TERT. And the reason that we had to get an immortalized cell line, as you can imagine, is that most normal cells will senesce after a few weeks or months. So we wanted to culture these cells in chronic alcohol, and we needed something that would not senesce. So they're about as normal as we could get. We cultured them in 30 millimolar ethanol twice weekly. And again, that's the work of Stephen Brown and Kelsey Roder-Gordy. They were cultured for 3 and 6 months. And from the 3 and 6 months time points, we harvested cells for ataxic analysis, RNA-seq methylation analysis, and whole exome sequencing for mutations. Now, 30 millimolar correlates to a blood alcohol value of 0.11%. You're considered legally impaired at 0.08%. So it's just a bit above legally drunk type of concentration. So it's a bit higher, but it's not super physiologically outrageous like some studies you might see in the literature. So we didn't know what to expect. So we really wanted to kind of hit these cells with a strong amount of alcohol, like someone might binge drink or drink chronically, or alcoholics might experience. So I need to get rid of this bar at the top of my screen because I can't quite see my titles. Does anyone know how to do that?
Go to the bottom, I think, and there's a presentation mode. Would that do it?
Are you seeing my slides okay? Are they large enough?
We're seeing them. Mm-hmm.
Okay, well, I'll just, I'm not sure how to do it, but I'll just ignore it. Okay, so what we decided to do was to assay how alcohol might affect chromatin architecture and accessibility. We used ataxic. Ataxic stands for Assay for Transposase Accessible Chromatin with High Throughput Sequencing. And this is from one of Cornell Schubert's papers, so I borrowed it from him. The ataxic method relies on next-gen sequencing, library construction using the hyperactive transposase TN5. Basically, you have to isolate nuclei and purify chromatin. And then you treat the chromatin with a hyperactive transposase homodimer with adapters that simultaneously fragments and tags accessible DNA or DNA that's open. And then you purify these fragments of DNA and PCR amplify them using the tags that were put on by the transposase. And then this library undergoes next-gen sequencing. This was conducted at our core here at Hopkins. And then you get individual peaks corresponding to regions of open chromatin. And the reason I wanted to give you this schematic was to, as I talk about peaks in the next few slides, those peaks represent regions of open chromatin. So a little closer, waves, please. Like, I can't really see it. Put it right here. Thank you. So, so the HMEX exposed to alcohol for 3 months have actually been shown to have less open chromatin. And this table here on the left shows that the number of peaks in alcohol exposed in peaks in HMEX exposed to alcohol for 3 months. There are less peaks in those exposed to alcohol, and also less peaks those exposed to 6 months of alcohol. So there's less open chromatin in cells that were exposed to alcohol. Now, the graph on the right represents the peak coverage, and it's the percent per domain, and the chromatin was divided into kind of activity domains. And the peak coverage of the open chromatin was determined for each of these domains. And it was found that, again, there's less peak coverage per domain in alcohol-treated HMEX at 3 months and less so at 6 months. And so using kind of two different ways to analyze it, we found that alcohol led to less open chromatin. Now, why were the changes less at 6 months? It could be that, the cells are undergoing some kind of clonal selection, or they're adapting to the alcohol. But what was really surprising was the amount of less open chromatin that we observed at 3 months. Now, chromatin accessibility can be altered anywhere within a gene or in an intergenomic region. So we were wondering, where is this chromatin changing, and how might that affect gene expression? So this was conducted by, this work was done by Anuj and Mike Considine, and they looked at the peaks in different functional regions of the gene. And you can see here that we have different promoters from close to further out, 5' and 3' UTRs, exons, introns, and intergenic regions. So what was the most stunning, really, was that alcohol at 3 months, of the remaining, of the amount of open chromatin that was still present in these alcohol-treated cells, it seemed to shift more towards promoters. It's more promoter-centric. So it's maybe 6% more open chromatin found in promoters with 3 months of alcohol, and just maybe a couple percent more at 6 months. And this could be really significant if there's more open chromatin in promoters. What are those genes, and what genes are being expressed? So the ataxic summary of what alcohol is doing to chromatin at this point in our research is that exposure of HMEX to alcohol for 3 months seems to lead to less open chromatin across the genome. None of those domains that I discussed were more enhanced than others. It was kind of evenly spread out across the whole genome. And alcohol exposure also seems to lead to shifts in the distribution of the open chromatin across the structural units of genes, with more open chromatin being localized to promoters. Now, along with the chromatin work, we wanted to understand how methylation was changing across the genome as well. And so we did this work with the Illumina methylation EPIC 850K array. And again, this was done in our ECGC core. And it's a genome-wide DNA methylation analysis based on bisulfite conversion and Illumina technology. It quantitatively detects the total methylation level of over 850,000 methylation sites throughout the human genome at single nucleotide resolution. So when the DNA is bisulfite treated, unmethylated cytosines, both 5-methyl-C and 5-hydroxymethyl, are converted to uracils, while the methylated cytosines remain unchanged. The amplification that then occurs converts the uracils to thymidine. The Infinium technology interrogates these different differentiated loci using site-specific probes. And Cornell has reminded me that in our core, the Infinium 2 probes are used in this system. So you have probes for unmethylated and methylated loci. And that will help us to know how much is actually methylated in the genome. Now, Mike Considine and Leslie helped prepare these beautiful heat maps showing the probes that were altered or differentially regulated after 3 months of exposure and 6 months of exposure of these HMEX. And we had 4 independent replicates for the MOX and the treated. And you can see that there's significant shifts in methylation in the alcohol samples in both, going up and down. In fact, Leslie and his team commented that they've never seen such dramatic shifts in methylation. So why so dramatic? I don't know. There could be lots of causes. There could be DNA damage going on and all kinds of things changing, but the methylation shifts were significant. Now, I wanted to know whether any of the DNA methyltransferase enzymes might be regulated. And the answer to that is no, unfortunately. But what we did find was that there are some epigenetic modifiers that are regulated by alcohol at both 3 and 6 months. Now, these are not known to be epigenetically regulated, so they might be being regulated transcriptionally, but these could all play a role in chromatin modification. And these are just some of the epigenetic modifiers that were regulated. These are the ones that are in common at both time points. The genes represent 15% of the differentially expressed epigenetic modifiers for 3 months and 36% of the differentially expressed modifiers for 6 months. The direction and magnitude of regulation is similar at 3 and 6 months for all cases, which was pretty amazing. And these genes are not known to be regulated by methylation, as I mentioned. Could they be playing a role in chromatin architecture? You know, who knows? Now, we want to understand, of course, now that we're getting information about methylated genes and genes that are expressed, are they similar to cancer or some kind of pre-malignant states? So what we did was we were aware of Chris Umbrecht has a really great DCIS data set that Leslie and his team had been working with. And I wanted to just introduce you to a little bit about what DCIS is. DCIS is, it's a breast lesion. It's a ductal carcinoma in situ. And it's represented here in this middle panel. It's basically a group of cancerous cells which remain in their normal location. They're within the duct. They haven't invaded through the basement membrane. They haven't become malignant yet. They're basically stage 0. DCIS develops through multiple stages. You know, the lumen is usually a single layer of luminoepithelial cells, but then you can get ductal hyperplasia through different reasons. Ductal hyperplasia might sit there as itself, or it might advance to atypical ductal hyperplasia. Then if there are more injuries and epigenetic changes and mutations, that could advance to a more carcinoma-like condition, which by itself, if it stays within a duct, is not so dangerous. But if it invades, microinvasion, then you can get development of breast cancer. So here are some human images showing what it looks like to a pathologist. And here is an example of a duct with microinvasions leaving the duct, becoming more dangerous for the woman. And so often in a mammogram, you'll see DCIS associated with calcium deposits. Now, in talking about these ductal growths, I also wanted to bring up a really exciting assay that we're doing with these these alcohol-treated HMECs. So the mouse has 10 mammary glands. There's 2 here. These are the number 5s. These are the number 4s, the inguinals. It has number 2 and number 3 thoracic glands, and then there's some up here by the salivary glands. And this mouse that's open here, you can see that the mammary glands lie just below the skin. They're just little layers of fat and vasculature, and they're not, you know, they're just laid against the body cavity. And you can get to them very easily. You can inject cells right into the mammary fat pad, or you can inject them into the nipple, intraductal injections. If you really want to mimic an atypical hyperplastic kind of lesion or a DCIS, you know, pre-malignant cancers, this is the way to do it. So this is work that we're doing in my lab, and this would be a mammary duct that's been injected with a blue dye, and you can see what the duct looks like in the mammary fat pad. Mouse nipples only have 1 duct associated with them, as opposed to humans who have maybe 5 or 6 ducts associated with them. And then, you know, what we plan to do is inject these alcohol-treated HMECs into these ducts to see what the biology is, to match that with our bioinformatics data. And you can see we might see some types of DCIS or carcinomas that look like these examples. And I am recruiting postdocs for this, so I'm shamelessly recruiting on this slide. If anyone's interested, please let me know. Now, this graph here represents the methylated probes in the DCIS samples that I just talked about that Chris Umbrecht has, as well as in our alcohol-treated HMECs. And as Leslie has explained this to me, the accumulation basically you put a bar where you have genes that let me see, I can't see my screen at the top, but you basically put a bar where the probes overlap between the DCIS and the HMEC data sets. And the accumulation of bars at the ends means that the DCIS associated probes are disproportionately altered in the HMEC data. And the curved line above is a representation of the density of those lines. So there's more density here in the hypomethylated probes and also more density in the hypermethylated probes, showing that there are some probes that seem to be behaving the same in both data sets, the DCIS as well as in the HMEC treated. This was really pretty exciting. We saw it at both 3 months and 6 months. So the methylation profile, there's some similarities between alcohol treated human mammary epithelial cells and benign breast disease or potentially DCIS in women. Now, I also wanted to kind of take you through how Leslie and Mike do some of this methylation analysis, because as a biologist, I find it pretty cool. You know, we really do speak different languages, and I've been trying so hard to working with the bioinformatics to understand how they interpret the data and what it means. So I'm going to just kind of go through one example of what they do for me. So they took these differentially expressed methylated probes that we found in the DCIS as well as in the HMEC data. And in the differentially methylated 3-month alcohol versus mock samples, there were 85,000 probes that correlated to 19,000 genes. And in the 6-month data, the 6-month treated with alcohol versus mock, there are 112,000 probes and 20,000 genes. And then in the DCIS, there were 65,000 and 17,000 genes. And this is when they were looking at all probes. But then we wanted to really understand if a gene is truly differentially methylated. So we started looking at more of the promoter probes, which are going to control gene expression. So Mike's criteria for whether a gene is differentially methylated was that at least 25% of the promoter probes for a gene had to have statistically significant beta values. And the beta values are the amount of methylation, or it's the amount of methylated probes. And all the promoter probes that were statistically different for a gene had to go on a I don't know if that's statistically different or statistically significant, but they had to go in a uniform direction. So all the probes for that promoter had to be had increased methylation. You didn't want one with methylation going up, one down, one up, one down. So they had to be all going in the same direction. And so he then came up with that there were 5,496 genes in the 3-month, 7,800 in the 6-month, and 4,400 in the DCIS that seemed to be really methylated by either alcohol or the DCIS condition. So we wanted to know how many of these genes were in common between the DCIS and the alcohol treated HMECs. So in the 3-month, there were 901 genes that seemed to be methylated in both the DCIS and the 3-month treated HMECs. In the 6-month, there seems to be a bit more, and there's 793 genes in common between the DCIS and the 3 and the 6-month methylated HMEC samples. So these genes are really going to be interesting to understand. These are genes that are potentially epigenetically regulated in DCIS and in alcohol as well. And so this is something that we've just uncovered, and we're going to be pursuing the importance of these genes in the next several months. Now, Leslie has taken some of the downregulated genes that a subset of them that are most likely methylated, and he put those through a GC analysis. And what was really interesting was that some of the most significant gene sets were UV response damage, which, you know, I guess you might expect that with a DNA damage that's going on in these cells. But interestingly, estrogen response, androgen response, EMT, you know, these are breast cancer cells. And so I thought it was really interesting that EMT comes up a lot when we, in our analysis of these cells. But estrogen and androgen, I guess you might expect that because they're human mammary epithelial cells, but I thought that was very interesting. And And genes that are upregulated, and so maybe have decreased promoter, we also found that EMT was a pathway that came up. So and some of the cells that we've actually looked at under the microscope do seem to be more fibroblast-like, maybe more a little bit spindly. So it is interesting that EMT might be one of the results of alcohol treatment. Now, we do have gene expression from our RNA-seq data of our HMEX. We have differentially expressed genes. But unfortunately, the DCIS data set does not have controls for expression. They don't have the normal breast tissues. They just have the DCIS. They did have it for methylation, so we could do differentially expressed methylation analyses. But for the expression data, although the HMEX, we have differentially expressed data, they don't have that for the DCIS. So Leslie had to do a single sample gene set analysis to try to understand what similarities might there be between alcohol-treated HMEX and the DCIS genes. So differentially expressed genes from the 3-month alcohol-treated HMEX that were downregulated are shown here in the blue line, and upregulated are in green. And black lines represent this random set of DCIS genes that carry a proliferative signature. So in his analysis, he took random genes and kept running them through to see what kind of density plot he would generate from, just kind of random genes and luck almost. What are you going to see if it's by chance? And so the parts of the blue and green lines that fall outside of the black lines show us that the alcohol gene sets, both the blue and the green or the up and down regulated, are more involved in DCIS than random gene sets of the same size on account of the large tails that exist on both ends. So these are the genes that are similar, but not just by chance. It's probably something real going on there. So Leslie looked in the DCIS samples that progressed onto invasive cancer in Chris's data set, as well as those that did not progress. And again, the blue set has lower expression after treatment and the green has higher expression after treatment. Now, the plots show us that the blue down in alcohol genes are slightly lower in progressing DCIS than in non-progressors. So these are the DCIS genes. Let me think, is that right? Is that right, Leslie? I can't see my my title.
These are your alcohol. These are genes that were altered after treatment with alcohol for 3 months. And the genes that changed after 3 months of alcohol treatment, whether they went down after alcohol treatment, which is the blue on the top, or whether they went up after alcohol treatment, which is the green on the bottom. The same genes sort of generally, sort of weakly showed the same trend in DCIS progressors versus non-progressors. So alcohol treated samples looked more like DCIS progressors, and the mock treated cells that had not been exposed to alcohol looked more like DCIS non-progressors. These results, by the way, are not statistically significant, but I think it leads to some interesting hypotheses.
And it's thank you, Leslie. This is data just hot off the press, and I'm still getting my head wrapped around it. But DCIS samples and data sets are very, very tough to generate. A lot of them come from paraffin embedded sections, and they're very difficult to get DNA and RNA from. And so we're lucky that Chris has the data set that he has. Leslie also did analysis showing that the HMEC genes that are regulated by alcohol do seem to maybe have a weak association with disease severity. So you can see in this data set that the genes that went down in our alcohol treated cells correlate with basal and luminal B gene sets that also went down. And then if you look at the gene sets that went up in the HMEC genes treated with alcohol, here you see that there's a slight increase in the basal genes that went down with the down genes. Now they're going up with the up genes, and same with the luminal B, they're going up with the up genes. Again, just showing that there might be a association with disease severity. But you know, the analyses that our team is doing is every time they run something, it is showing, you know, it may be weak, but it's clearly showing an association with DCIS. So I think that that's really, really exciting and something that we need to pursue further with Chris and our bioinformatics team. And let me just give the summary now of what we have found thus far with our, you know, trying to understand how the epigenome is altered by alcohol. We found that there seems to be less open chromatin after 3 months of alcohol exposure. We're actually redoing this now to look at 1 month and earlier time points. There's a shift in the distribution of open chromatin to promoter regions after alcohol exposure. It'll be interesting to understand which promoters these are and which genes are being regulated. There seems to be really significant changes in DNA methylation after both 3 and 6 months of exposure to alcohol. Similarities between the methylation profile of alcohol exposed HMEX with the methylation profile observed in DCIS. So the probes are similar. We know that there's definitely some methylated genes that are similar. And there's a group of 700 or so differentially methylated genes that are in common between the alcohol treated HMEX and DCIS. And then in those last few slides that Leslie explained, there's differentially expressed HMEC genes that appear to overlap with genes in DCIS that might be associated with disease severity. So I hope I've convinced you that there's a link between alcohol consumption and breast cancer risk, that we know that alcohol can affect the chromatin accessibility, that it can affect massive changes in DNA methylation. The DNA methylation, of course, is going to change and alter gene expression, and that there may be some similarity that we're seeing in genes that are expressed in ductal carcinoma in situ, and maybe we'll even look earlier than that at some of Chris's more hyperplastic gene sets to see whether there's also similarities between the atypical ductal hyperplasia that precedes DCIS. And so with that, I'd like to thank you for your attention, and I'm happy to answer questions.
Great presentation, Cindy. It's great to see all this data coming together now. So, you know, as a full story now. I had just 2 quick questions. So when you expose these cells for alcohol, I know that, you know, you showed this data for long-term exposure. If you were to take alcohol out in any one set, and do they revert? Would they revert back? Not in the analysis sense, because of course methylation, ATAC, you know, those are all, you know, big stuff, just by looking at them. Because I think when you showed the first results, they were actually looking very different from, you know, from the unexposed cells.
Mm-hmm. Yeah, Dipali, that's a great question. And actually, we talked about that early on. I mean, I think we've been doing this project for 2 years now. So I did do that. I had some cells that were exposed for, I don't remember if it was a couple months or 3 months, and we withdrew alcohol. And we have, we did RNA-seq and methylation analysis on those. Oh. And they were still very, very epigenetically altered. They maybe weren't exactly the same as the continued treatment, but they were still different than the controls. And I actually had talked with some colleagues at Texas A&M who were doing that work as well. And I withdrew the alcohol long enough that you should see complete reversal if it was going to happen. And, you know, a lot of these methylation changes, they're hard to reverse. You know, they get pretty locked in. So that's a great question. We did see some reversal, but it did not go back to baseline.
All right. And if you were to, I think you might be actually doing this now. Do they make these cells, the, you know, normal mammary epithelials, as tumorigenic? So if you put these alcohol exposed cells to mice, are they forming tumors?
So we're doing that now.
Okay.
Well, I wanted to wait until we saw the bioinformatics data to know whether, you know, is 3 and 6 months enough? Are we seeing enough changes before we start putting them in introductorily? Because we're doing that, and we don't have the answers yet. We're hoping, you know, it's going to be a nice experiment because these are human cells into mammary ducts, so it'll be very easy to identify any abnormal lesions. So I don't have answers for that yet.
Yeah, I'm looking forward to it because your RNA-seq shows all this enrichment of these pathways. Looks like, you know, you might see something there in vivo.
Yeah, I know. And I would love if the breast group wants to help me with some of the injections.
Sure.
That would be great because we're
I
trying
asked
to.
my lab to be there on the talk.
Yeah, I would love to have help with the injections. My new technician is learning them, but they're quite, as you know, they're quite challenging. You can only inject about 2 microliters of solution, and it's done under a dissecting scope.
Yeah.
So, but I think that's going to be very exciting. And then we're also growing cells now. We're harvesting them at earlier time points, and then maybe even later than 6 months. So we'll have a lot of cells that we can put in.
Great. Thank you.
Sure. Now let me check out the chat. I think I saw a
Yeah.
question about red
Yeah,
wine.
Cindy, there's a couple of questions there. Yeah, I was going to suggest we go there.
Yeah, you know, that question comes up all the time. And I actually gave a talk down in Annapolis or someplace about that. Red wine still has alcohol. Okay, it's the resveratrol that have cardiac promoting, you know, health benefits. There are a lot of conflicting studies out there, but I think what I have to tell you is that there may be some health benefits of resveratrol, but you know, you can always just eat grapes too, or drink, you know, other, you know, grape juice, or if you really want to
Right,
get the.
but wine is more fun, and you know, since we're talking about, you know, alcohol, yeah.
I totally agree with you, and I have to tell you, this is really difficult to talk about this subject matter at some type of cocktail party. It's the real downer for people. I like wine and champagne and as much as anybody else. But what I can say is drink in moderation. You know, I mean, instead of having that 4th glass, just stop at 2 or 3. There is alcohol in wine that is that definitely can increase your risk for breast cancer. The studies have shown that it's not the type of drink, whether it's, you know, jungle juice with sugar, carbohydrates in it, or whether it's a cocktail with vermouth and vodka, or whether it's beer, they all can increase your risk of cancer. So it's truly the alcohol itself. So don't think that red wine is safe. Just drink in moderation.
Okay.
So Cindy, there's a question: would transgenerational epigenetic inheritance make offspring better tolerate the stresses of alcohol?
You know, that is another really important question that I don't think we understand yet. I think that it can make, I think that some of the studies have shown that it can make families of alcoholics, you know, more susceptible to alcoholism. In some Asian cultures, there are inheritance of enzymes that modify and metabolize alcohol. We don't know, I think, enough yet about the epigenetic marks in cancer. It does worry me, you know, as a breast cancer survivor myself. My heritage is Lithuanian, and I know that Lithuanians like to drink their alcohol. And did I inherit some epigenetic marks that maybe had me a little further along the pipeline to cancer than you know, other people? So I don't know what the results would be. I don't know whether it would protect you or make you more at risk. We don't know, but you can inherit these marks that might play some role.
And then another question in the chat. They said, I'd be interested about genetic markers for glycine methylation.
Yeah, I don't know that I've read much about that. That's an interesting comment. And yeah, I'm sorry, I don't know much about that.
That was from Greta Becker, if you, in follow-up, yeah.
Happy to talk more about that, right.
Any other questions for Cindy at this moment? Well, Cindy, I want to thank you so much for presenting today. You know, working in public health, I think this is a frontier. You might have seen the Baltimore Sun this morning had an editorial about a tax increase for alcohol.
Oh,
before the Maryland legislature right now. These tax bills tend to pass when there is a need for additional income at the state level, otherwise the legislature isn't interested. So I'd encourage anybody on this call, you as a private citizen can email your legislator. They're all on the, on the internet about your thoughts about the tax increase. We are doing some calculations about the changes, and it would reduce the cancers by about 1.5 to 2% in Maryland. Excuse me.
Can I make a comment? This is John Fetting.
hi, John. Sure.
Hey, Cindy, on behalf of our breast cancer team and the Fetting Fund, we're very happy that you're taking up this work. It has been as you pointed out, the epidemiologic data linking alcohol and breast cancer is very substantial and very solid. And so, there are probably lots of reasons why it doesn't impact health behaviors. But I think one of the issues has been that we've not really gotten beyond the association between alcohol and breast cancer. We've really not come up with mechanistic explanations for it. I think it's easier for people to ignore the alcohol if they think alcohol is a marker for something else. But I think to the extent that we can begin to nail down how alcohol does, over time play a causative role, I think that would be very important. I'm hoping the kind of work you're doing is in that direction.
Thanks for your comment, John. I think it's really, really complex question and literature, because it depends on how much you drink. If you drink 7 drinks a week, but they're all on Saturday, that's a very different exposure than drinking 1 drink a day. Also, your premenopausal status, your postmenopausal status, have you had cancer? Did you drink before your diagnosis? There's so many questions that make the literature a little bit confusing. And all the models are very different in how they treat cells or treat animals. So I do hope that we can start to get at more of the mechanism behind how in the world breast cancer can be influenced by alcohol intake. And I'm hoping that these DCIS data sets that we have might give us some leads on some of the genes that might be important. And thank you, John, for your support. I couldn't have done it without you and the bioinformatics team that were, they're expensive. So appreciate it.
We're glad to help.
Well, Cindy, I was going to ask you about obesity. Is there any way to evaluate, whether there's a joint effect of obesity and alcohol, or whether it's a direct effect of each?
Ooh, the obesity literature, I don't even want to, that might be DePauli can talk about that a little bit. Or there's metabolism, there's some metabolism that there's overlap with metabolism. But obesity also involves hormones, and there's a lot of complications there between.
Right.
I think, Norma, that they both are risks. So if you drink and you're overweight, you probably have an increased risk. I don't know if they synergize with each other or both contribute separately. But I do know, I want to tell you this. So I had, believe it or not, Glamour magazine had asked me a question they wanted me to answer. And they didn't want my pictures or anything, unfortunately, but they wanted me to talk about the relationship between why some groups of people, lesbians or transgenders or others, might have an increased risk for breast cancer. And what it came down to when I looked in the literature, and that's how I became aware of this link with alcohol, was that some groups of individuals tend to maybe frequent bars and socialize more in settings where there's a lot of alcohol consumed. And so the link with the breast cancer in some groups of people that are more in situations where there's a lot of alcohol and cigarette smoke, it was probably linked to the alcohol consumption, and also other reasons as well. But alcohol consumption and obesity are definitely gonna increase your risk for cancer.
Mhm.
Yeah, that's a complicated question.
Yeah, I'm not
Polly,
aware.
did you want to say something?
Yeah, I was just gonna add that I'm not aware of any study that has looked at the link between these two. But as you said, Cindy, they're both actually triggering different hormone, like different pathways. Because obesity, we know a whole lot about pathways that it's triggering, and alcohol, I didn't see any overlap yet. So maybe these are like, if you're obese and you drink, you are adding two risk factors. One thing I would like to add here is that I've seen papers that obesity changes your microbiome. It alters the microbiome, and now we are finding connections with microbiome changes and breast cancer. And similarly, alcohol does that same too.
Right.
So maybe with microbiome, we can find a link that if a person is obese and drinks, then there may be changes in the microbiome
Yeah.
that might enhance risks. But that's
And
like,
that's likely the mechanism is not going to just be epigenetics or metabolism or microbiome. And there's so much involved. It's going to be the balance of all those factors.
right. Yeah.
Yeah.
Okay, Cindy,
may-
we'll close for today, but appreciate your presentation. And I'll mention to the audience again, this has been recorded and we will post it on YouTube and let our listserv know when it's available. So thank you all. We'll adjourn.
All right, there's one more or two more comments in the chat. Greta mentions the CYP21A2 and also the I'm sorry, I'm not a scientist. I can't pronounce this. So I just wanted to let you know it was there.
Yeah, so the CYP enzymes, so certainly they're going to be affected by metabolism. I know that, as I mentioned earlier in the talk, there's a lot of effects on hepatocytes and metabolism. And glucocorticoids can definitely be affected because alcohol can suppress the immune system. And so I would imagine that cortisol and glucocorticoids could be suppressed with excessive alcohol drink. And that's going to create an imbalance too. So thank you all for your comments. It's a very complex exposure type of mechanism, and there's a lot involved, and appreciate you all for listening. Thank you.
Okay,
Thank you.
bye-bye.
Bye-bye.